For everyone with an interest in NASH, or more broadly, fatty liver disease, Surf's Up, Season 4, Episode 1 of Surfing the NASH Tsunami. Our look at the first day of NASH-TAG 2023 starts now.
Today, on a special NASH-TAG episode of Surfing the NASH Tsunami.
It's academically the challenge for us, or also the interest, to develop new NITs, and I think there's room for that, but we don't need more newer NITs. We can start treating patients now.
Designing cirrhotic trials, especially with entry criteria with NITs, is probably the easiest goal in terms of skipping biopsy.
If we just step back and look at what's being presented, we have clues to all of these phase 2s to help design the phase 3s.
Hope is a really big thing for people, and for patients it makes all the difference in the world to know that there's something coming.
To kick off 2023, over 300 attendees representing the range of fatty liver stakeholders have come to Deer Valley, Utah, or joined online for NASH-TAG 2023. Join hepatology researcher and key opinion leader Professor Jörn Schattenberg, liver wellness advocate Louise Campbell, pricing and forecasting guru Roger Green, and our guests, hepatology researchers and key opinion leaders Drs. Stephen Harrison and Mazen Noureddin, and patient advocates Mike Buttel and Tony Vigliotti, as they discuss what we are learning and how it will affect NASH stakeholders today on this special edition of Surfing the NASH Tsunami.
Hey listeners, welcome to the beginning of a new Surfing the NASH Tsunami season. It's Season 4, which I find amazing to consider. We start this season with 3 episodes from what promises to be an energizing and enlightening NASH-TAG, complete with new data on drugs and NITs, big answers to big questions, and, for the second year in a row, a regulatory fireside chat on Saturday evening. As Mike Charlton kicked off this morning's session with his annual year in review, he noted that he had considered describing 2022 as, and I quote, the best NASH year ever, but then thought about what 2023 would bring and change the title to the best NASH year in review yet. Seems totally appropriate to me. 2023 might be gangbusters. We'll be posting 3 episodes over the next few days, stemming from 5 recording sessions. Before I start today's recording sessions, let me take a minute to describe why we're covering this the way we are. For the NASH Tsunami team, there are 5 separate streams of positive activity going on here. First, some great drug trials, including the phase 3 results for resmetirom, the obeticholic acid reanalysis, and an array of positive phase 2 results. Second, exciting and important new data on NITs. Third, some tremendous breakthroughs in basic liver science. Fourth, perhaps some insights from ongoing regulatory discussions that can streamline the path to drug approvals, both in terms of time and budget. And fifth, in some ways most important of all, bursts of optimism from every key stakeholder in the space about the pace of progress and the high level of confidence that all this work is finally, clearly headed in the right direction. We will bring all 5 streams into our 3 episodes. Today's episode starts with our Saturday morning discussion with Jörn Schattenberg, Stephen Harrison, and Mazen Noureddin about all the Friday sessions, and that'll be followed by a discussion with Louise, Mike Buttel from the Fatty Liver Alliance, and Tony Vigliotti from NASH Knowledge about what the story of this conference, which neither Mike nor Tony is able to attend, means, and what the drug presentation suggests about how it will affect and change the nature of their work and energize what they're doing. Tomorrow's episode, which is sponsored by HistoIndex, starts with a discussion between Louise Campbell, Scott Freeman, and Donna Cryer, which will be 1 part basic science, 1 part overview, and maybe a little bit like the conversation the 2 of them had at NASH-TAG last year. Our second conversation will feature Jörn Schattenberg, Louise, Quentin Anstey, Rachel Zayas, and perhaps 1 more key opinion leader, focusing largely on tomorrow evening's fireside chats and general meeting themes. The final conversation from this episode will come from our sponsors at HistoIndex, and will feature HistoIndex Chief Scientific Officer Dean Tai, Jörn Schattenberg, and me discussing a fascinating poster that Jörn and Dean are presenting at this meeting on how AI can clarify drug effects in different zones of the liver. I consider this to be extremely promising for drug development as we move past the does it work phase into the how does it work and where can it fit best into drug therapy phase of advanced drug development and treatment strategies. Monday, we'll have a diverse set of stakeholders joining us to consider what we will have just learned from a range of perspectives and all various presentations. And with that, let me stop and we'll start today's discussions. So, as I just mentioned in the introduction, I've decided to change the order of today's episode, start with the folks who bring the heft from yesterday and then go later to the patient advocates and what they had to say. So this morning with us we have Stephen and Mazen and Jörn. Stephen, congratulations on a fantastic conference.
Thank you. So far, so good.
Yeah, and whatever you did to talk to the weather gods about bringing the snow, of course you did that the year I wasn't there, but I don't take it personally. I'm sure you had another motivation in hand. Maybe when you get Quentin and Phil out on the slopes later for their ski lesson.
I think it's just parallelism, symbolism, whatever you want to call it, the depth and extent of the knowledge that's being, handed out at this meeting is paralleled by the depth and amount of snow that's falling outside.
For those planning to attend NASH-TAG 2024, Dr. Harrison just advised you to ski in 2 days early and plan to fly out 3 days late after the airport gets snowed in by the 4 feet of snow they're going to get, but because that's the only way you top this. But Jörn, good morning. How are you?
Good. I'm here. It's a real pleasure to attend in person. Congratulations to the course directors and Stephen for setting this up. It's really a treat and a great experience.
And finally, Mazen, how are you this morning, Mazen?
Good morning, man. As you know, it's 6:15. There's a bunch of snow outside, and it's beautiful here.
So good for that. For those who heard Mazen's session yesterday afternoon, I did try to get somebody with an English accent onto this because he alluded to the idea of being the only non-English accent in his section yesterday afternoon. But you're just going to have to be the best we can do, and that's a German accent. But
I'll do my very best.
Cheerio. So with all that, why don't we just get started? Simple icebreaker. The one thing that you saw or heard yesterday that you think had the greatest impact or made the greatest impression on you. Brave one, go first.
I'm brave. I did a little skiing yesterday, which was tremendous. But the icebreaker, or the most important thing for me, was I'd like to revisit the statement that Phil Newsom made, and that was the session Mazen and Naeem Al-Khoury chaired, which was great, I think. Sometimes we're looking at NITs as the absolute truth, and they're not. They're helping us to refine the patient that's sitting in front of us, or more specifically, refine the management pathway. And I think it's something that's going to be very important moving forward and assessing NITs, in particular if we move away from the biopsy in coming to an endpoint.
Okay, I'd love to come back to that because that statement, I also took a note on that statement. In part, Jörn, in context of the interview that we're doing with Dean Tai in tomorrow's episode, that there are going to be so many ways to look at this stuff and so many pieces that can make contribution.
It's hard to come up with the one thing that stood out. I was thinking about that this morning in preparation for this meeting. I don't have one thing. One thing just doesn't come to mind. There was a lot of discussion around the mechanisms, around some of the exciting new therapies that were coming out. Honestly, for me, the most exciting moment was giving Brent Tetry his mentor award because he was my mentor. So it was great to see him be a part of the meeting and to win that. But as far as the content goes, I guess I don't have one thing that stands out. There's a lot to talk about. I'll say that.
Okay, so I will note that Dr. Harrison, when presenting Dr. Tetry's award yesterday, noted that it took multiple mentors to get him through. So I'm wondering for how many years we're going to be doing your mentors. But I think that I agree with you. Fantastic guy and a really important contributor and a great choice.
Well, look, Scott Friedman was there. Scott Friedman and Brent Tetry chaired a session. Scott was Brent's mentor. So it's like we have the grandfather of mentors. I don't know how many generations we can take this thing.
Well, Scott and I are roughly the same age, so I'm starting to feel really old when I hear you talking about being the grandfather of 50-year-olds, but that's a good thing. Okay, Mazen, what was yours from yesterday?
I didn't know how old you are, Roger, but anyways, that set the stage for what I'm going to say. I think I'm going to take a little bit different angle, and what I liked, and I heard that comment from multiple people, is the culture and the atmosphere of NASH-TAG, which is very relaxed, friendly. A lot of people kept saying, you guys know each other. It's kind of like formal and not formal at the same time. People come in skiing clothes, or they come like an outfit like Stephen Harrison, like he looked like Zelensky's, President Zelensky. You can pick on Stephen Harrison and tell him he's eating burger all day. Rohit is picking on Vlad that he went overtime, and then he goes way more overtime than him. And it's just like fun, and people are friends and, having great time. So that's what I like about NASH-TAG.
Greatest compliment this podcast ever got was somebody once told me that it felt slightly like NASH-TAG in exactly that way. So I couldn't agree more. I just think it's a fantastic, as you know, I'm sorry not to be there.
Yeah, this year I'm enjoying picking on Steven. I know I'll pay for it later. I might not get invited next year, but it's fun, man.
No, he's just going to come up with a better math test for you to do than you came up with for him yesterday. That's what's going to happen. He's going to find math maybe you can't do.
That was the red line.
How am I supposed to track a Fagan nomogram and come up, looking at a post-test likelihood ratio? And on the spot. Come on. That was only 9% off.
That was great.
Yeah, so first of all, the phaga nomogram might have been mine. I'd never seen that before yesterday, which shows everything I don't know. But I just really like the elegance of that, number one. My second takeaway was, Mazen, no matter how many times you talk about the idea that what we're really looking for is correct total predictions, that's another one of those things that I think is just a lot more elegant than the statistics that people use to get there. I mean, we throw tons of numbers around, but the question really is, what are you trying to see at the end of the day? I think that's a really good example of cutting to the core of what matters, phaga nomogram being second. And the third, Stephen, I'm not there, unfortunately, but this is the fifth one of these I've seen, which is kind of frightening if you think about it. The questions get better every year. Not only does the data get better every year, but even more important to me, the questions get better every year, because the questions are what prove to me what have you learned and what haven't you learned. You know, there were just so many of them.
Well, I would say some questions get better. Some I was a bit frustrated that they kept asking the same question. And the one that I think we need to put to bed is the liver biopsies are not going to be required to put somebody on drug. I don't know why that keeps coming up. We don't do liver biopsies anymore. So why would the payers expect us to do liver biopsies? I think that is one of the definitive statements we can make. We're not going to do liver biopsies when drug therapy is here.
I agree. And I think that statement came across strong. The question was out of the audience. Yeah, it felt like it was not very medically orientated, the question. So I'd like to underline that. And my statement just before I hand it over to Mazen is, I didn't mean to implement we can't use NITs. We will use NITs to assess the patients. It's just there's things that influence the reading of the NIT. I think that's what the phaga nomogram tells us in the end.
Let me just look at this the way I looked at it. There were questions that I agree with Stephen that they came out kind of old and repetitive, but nevertheless, the glass is half full. We had multiple investors in the room, which shows you that there's a huge interest back in NASH. And all along the way, we were saying, this is going to work. We're just working on it. Look at the Hep C, it took longer. And here we are, we have 2 positive phase 3 studies and potential 2 drugs to be approved in 2023. And what we're hearing at a certain point in this journey that, well, NASH is risky, NASH is this, NASH is that. And we have seen this year that what the experts and others said all along, that this is going to work, just be patient. So we got a lot of questions from the investing community, which shows the huge interest back in NASH from that community. So despite some questions like the biopsy one, I know the payers would not ask for a biopsy. Indeed, they have mentioned that to us multiple times when we sat down with them on the same table. But I'm glad to see hopefully more investment in NASH that will support our patients at the end of the day.
So not being in the room, Mazen, I kind of had the same take. Stephen, I think as the disease grows in momentum, this meeting will get bigger. It's getting bigger. As it gets bigger, the people who are going to come are not going to be the people who've been along for the entire ride, but the people who are showing up later. And they'll have less knowledge and, frankly, their own biases. And if you're an investor, the first question you always ask is about payers. When I wind up talking to investors for people, I spend more time talking about what a payer is going to do than anything else. And I am struck by how misinformed investors are about they view payers as being very one-dimensional, and they're not. Now, that being said, I had exactly that same debate at this meeting 3 years ago with a leading KOL who told me that the idea that payers weren't going to require biopsy for everybody was misinformed and just didn't understand payers. And that was somebody who I think probably would be considered in the top 20 U.S. people. So now the top 20 U.S. people know better, but that misinformation has kind of permeated more broadly.
Just speaking about another question that I got that was interesting, I think it spurred a bit of good discussion, and it'll be maybe debated a bit more today, was the one with resmetirom. And Naga Chalasani asked a provocative question about why don't we just use thyroid hormone replacement therapy? And, if a THR beta agonist works, why not just use Synthroid? Now, I think he knows the answer to that, but he's putting it out there as a question because it was raised by one of his fellows or junior faculty, and he wanted to have it debated and discussed. And I think the take-home message there is when you have a hypothyroid liver, giving Synthroid doesn't fix that problem. It just adds more reverse T3. But systemically, you're going to see the effects of too much thyroid hormone acting outside of the liver. And there will be a very good discussion today by 2 endocrinologists in one of the afternoon sessions. I think it's one of the sessions like you did yesterday, Mazen, with Intercept. Madrigal has their session today, and Evelyn Broomstreet from Amsterdam, I can't remember her last name well, but she's going to be talking in detail about THR beta and its effects on the liver. But I think that generated some good discussion discussion around mechanism and what's really happening with that drug. I think there's just a lot we don't fully understand in the liver around how that drug is working, but I think it generated good discussion.
She's really an expert on the thyroid hormone metabolism, and I heard her talk once before. It's very interesting. We'll learn about target engagement also and potentially drug response, because that's important for us too moving forward. So I think the whole discussion around the sex hormone binding globally and how is that linking into this treatment paradigm, and of course reversing the hepatic hypothyroidism, which comes down to the questioning that Naga posed is you got to fix that and not the systemic problems.
Let me make a really quick comment on these kind of talks. I was actually surprised about the amount of people that showed up to the Intercept Symposium, not because of anything, because this conference is very specialized, target drug therapy, but sessions like this, there are people that, as Roger said, they're just walking in, they're just starting. So we are going to a lot of basic things, talking about the disease, prevalence, the component of fibrosis, and a lot of people came up and said, well, that was very informative, which I did not expect. But the point is, in NASH-TAG, I think there's, congratulations, guys, you have a wide range. We had NITs talk that we talked about likelihood and math, we talked about AUCs, we talked about correct classification, but also there's the other spectrum that covers the disease from A to Z. And for those that they don't know the disease well, if they come here, they will learn about it as well.
Yeah, I think that's right. So I observed something similar, gosh, it must be a decade ago now, with the meeting on immunotherapy in cancer, which started out as an academic meeting where people got to kick around big ideas. And over time, starting with the approval of Keytruda and Opdivo, morphed into a much, much bigger and broader meeting, ranging from a lot of financial analysts to drug companies trying to figure out how to keep the basic science in the middle of the meeting. I think, Stephen, frankly, that's going to become a challenge for you guys as time goes on. And what you're seeing right now is the beginning of it. Yeah, Mazen, that doesn't surprise me for the reason I just gave. And I expect that when we come back in 24, there will probably be more people wanting to do those events during the afternoon, and there will be more of an audience for them, and the audience will probably be as mixed, maybe naive, as the one you were looking at yesterday.
Not naive. I mean, they're all smart people. It's just like different stage of entry. But good luck finding time for skiing, Stephen. You guys have to. It's getting better and better. So the challenge also the skiing time.
Yes, Stephen.
Yeah, there is really no ski time for me till Sunday. It's gotten that way, but that's okay. I mean, one of the things about NASH-TAG that we don't really talk about a lot is just the networking. We all go to AASLD, we all go to EASL, but I don't know what it is. Maybe it's so spread out. Maybe it's people are a bit more insular. But I think at this meeting, people let their hair down a little bit, and everybody's kind of close-knit and connected, and there's just lots of opportunity or maybe they let their hair go. I don't know. Just whatever.
Well, I echo that. I think the networking is the best part of this. Anyways, go ahead, Roger.
For those of us, you don't know because you're on audio, not on video, but at least two of the four of us have hair that could really use work right now. And the other two of the four of us, being Dr. Harrison and myself, don't have that much hair to work on anyway. So it's a whole different kind of question.
It's an early morning hour.
Yeah, it is. Let's go back to the scientific heart of the meeting. What came out of this meeting yesterday that you think was science that people need to understand better or differently because of the conversations? It's not exactly how I wrote the agenda, but I think that is the question.
Let me start from my session and some of the comments that I heard. I had someone from GSK, who very knowledgeable guy, came up to me and he said, well, I looked at the NIT session and I said, should I go or should I not? And I'm glad I came. I thought it will be the same old thing, telling us about fast and mast, but it was really, in my mind, I mean, I'm biased because I was of this session, but I was very pleased to hear what Nimble and Litmus have done. So now, I'm summarizing my session. I think they contributed to the field significantly. Arun Sanyal talked about a couple of things. The two things that they strike my mind is how he showed up multiple tests and how they perform. So they actually are producing now, and he showed us how they're testing, for instance, imaging, and look at the repeatability of imaging markers across centers. Then Quentin came and talked about Litmus and the huge number of sample size, in addition to them tapping on multi-aspects, including machine learning, diagnosing the disease, and eventually they're going to start, or they already started collecting outcomes. I was very pleased to hear that. And then with the next three talks, I think think what we tried to do is we had NITs all along the way, including we started from FIB4, moved to transelastography, and then MRE came. But what I was noticing that people were getting a little bit NITs fatigue, that there are too many of them, despite the fact that we had multiple good biomarkers and scores that came up in the last 2 years after the pandemic, including MAS, MAFib, NIS4, NIS2, MASSIVE, you name it. So it's like going to perfection to pin it down. And I go back to the statement that Jörn mentioned about Felnusum. They're not perfect, but we're getting closer. Some people said, well, you guys keep moving fast and quick, even with NIT, with the target. So I think that session highlighted all the new highlights in the world of NITs on both sides, how to use it clinically and in the patients in clinic as well as in clinical trials. And the context of use is very important. You can switch between sensitivity and specificity, depends where you are, in office or you're screening for clinical trials or you're looking for treatment response. So personally, I was quite excited about it.
Mazen is very smart. He understands all these different sensitivity and specificities. I think you had 7 terms on your list, including the correctness, right? Which one was that? The correct classifier. Apologies.
Classification, which is like what gives you money for your pocket, right? That what is actually you're really detecting true with your NITs.
I think bottom line here is we have enough NITs to treat patients if a drug becomes available, decide about treatment response. We're going to learn more from the corresponding trial. It's academically the challenge for us, or also the interest, to develop new NITs, and I think there's room for that, but we don't need more newer NITs. We can start treating patients now.
Jörn, it's an interesting point that the two of you make collectively. If NITs follow other technologies not in medicine, and even in fact in medicine, what happens after a while is that something is good enough and broad enough and gets enough commercial momentum behind it that it tends to take over. And at that point, even better tests have a hard time making their way in, because sometimes we decide that we've gotten to good enough and then we just build on the platform we have. The most famous example of that, well, there are bunches of them in technology, but why are we all using Microsoft instead of Apple? Because 40 years ago, some guy who developed an operating system for Apple didn't want to talk to IBM. They wound up with Bill Gates instead, just a fluke of nature. At some point, we're going to get this right enough for treating patients that's going to become the platform, and people are going to move on to other questions. Not entirely. They'll keep developing, but it's going to take a much bigger threshold to get over that. And frankly, Mazen, not just for clinical trials, but I wonder if correct classification, even for patient treatment, is going to be the hill on which NITs wind up living and dying. Because at the end of the day, right, the goal is when you look at a patient, how quickly can you figure out what's really going on? And in that moment, indeterminate zone is of no value at all. So the more you talk about this, the more it hits me that might actually be where the solution winds up lying for everybody in what's the most efficient way to get to a correct classification.
Well, I'm biased in one way or another because I was fortunate to be part of creating a test that had a very low indeterminate zone, which is the MAST. But the other tests can be mitigated by, as you know, sequential testing and narrowing down that indeterminate zone, though that will cost more because you're doing another test. But yeah, I have to give a credit to Laurent Costera, actually, who is probably the most knowledgeable about correct classification or true classification, whatever you want to call it. And I had many discussions with him about that.
I think the whole NIT session that we had was fantastic yesterday. We'll have an extension of that today where we begin to look at these NITs with specific contexts of use. But it's evolving, it's being refined. And I like the way Mazen took a slide out of a different therapeutic area and said, it's not just a challenge in NASH, it's a challenge in multiple other areas. I think he highlighted Alzheimer's as one of the challenges in building a myriad of non-invasive tests to help refine who should get therapy and how to monitor response. And that was actually apropos, knowing that a drug got approved for Alzheimer's yesterday. So I don't know, you foretold something there. Hopefully that'll continue to evolve in our field. And I think if you give a talk on NITs, there's so many of them, you think you're going to forget one. As you begin to put the blood-based biomarkers, the proprietary biomarkers that blend several together with the imaging biomarkers, and then the combination of imaging and blood-based biomarkers, I always think, which one am I forgetting? There's a whole bunch of them out there. But I think it's like anything else. We'll learn how to use them. We'll refine them. We'll figure out how to incorporate likelihood ratios and Fagan's nomogram or however we want to do it. It's great to see that evolution of discussions around NITs, where maybe 7 years ago when we launched NASH-TAG, it was, well, here's an NIT, and we're nowhere near the level of knowledge that we are now. So, Mazen, I challenge you to continue to refine your work in the area, because 7 years from now, we're going to look back on this meeting and say, wow, remember where we were back in 2023?
And now we have drugs that they're working, so we can look at the response to therapy. So I think the way I see to that, although we really want to test NITs against outcomes, which as multiple people said, this is probably the end game goal or the most important part. I think now the next step with these efficacious therapies that we look at NITs correlation with histological response, while we're building things like male NITs and other consortiums and trials that have outcomes and look to the correlation with NITs and outcomes. And I'm sorry to hijack the beginning of the talk with NITs, but I really also want to discuss the next session that Stephen, Rohit, and Vlad did. So I love that session, to be honest. I'm glad that Vlad went over his time because he gave a brilliant talk. And as I said, Rohit picked on him and because he went over his time. And Rohit went over his time and also gave an outstanding talk. And then the finale was great. Stephen came and talked about cirrhotic trials, which is something we really need to talk about more because now we know that we have efficacious drug. We know from the hepatology literature that cirrhosis is reversible. And there's a proof of concept study that fructifermin might work on cirrhosis. So I think we're moving the needle there. So starting with Vlad's talk, I think he has a review paper coming in hepatology, and I think he used many of the slides from that paper. It was just brilliant talking about why the combo are important, the challenges why we have with the combo, considerations with the combo therapy, and I'll let others comment on it as well. But I thought it was very well done. Rohit's talk was also outstanding. He did not complicate it with digging deep to all the SNPs that we know about and rightfully focus on the two most important SNPs that they're most promising for therapies, PNLP3 and HSD. I thought his approach is interesting, like his futuristic look that you look at HSD and PNLP3 and you do precision medicine, and if the patients don't fit that, you can move them to other therapies. I might look at it a little bit differently from that perspective. I probably will combine the first talk with the second talk, and I say we might genotype patients eventually, and the combo drug will be based on the genotype that they come up with, give them one precision medicine drug and another drug that are efficacious, maybe the precision medicine drug as induction therapy and the other as a follow-up therapy. So there are multiple ways to look at this. And finally, the king of clinical trials design topped that at the end with the cirrhotic trial design. I think actually, to be honest, designing cirrhotic trials, especially with entry criteria with NITs, is probably the easiest goal in terms of skipping biopsy. And we started seeing that with the resmetirom outcome data, that we don't need a biopsy within 6 months of the trial. It can be as long as 5 years. Indeed, I hope that they get rid of the biopsy completely based on the liver form criteria that we put together, but I think they were still cautious and they put biopsy up to 5 years. But I think cirrhotic trials can be a great entry for get rid of the biopsy completely, look at outcomes. We're very sure now how to diagnose cirrhosis based on NITs. I can talk way more, but I want to leave the chance to the others.
Mazen, I'm with you. This is important. I think this is really where we're going to see outcomes. And I think we've discussed this before on this podcast. Now, the other thing that maybe I was wondering whether you heard an answer to that is how do we decide when patients actually become cirrhotic? And there were some numbers we're thinking about continuing them in our trials that we enrolled them in F3, and then we have to follow them up and maybe decide when they are cirrhotic. I think this is like the next arena where NITs will be challenged to really depict those patients because they develop outcomes. I mean, the discussion around the progression to cirrhosis is relevant, but we need something to follow them.
Your comments made me think of another point that's coming out of this meeting, and my days are mixed up a little bit. I think a lot of it will come out today, but the way that these NITs are starting to foretell in a much more accurate way than liver biopsy what's happening with drug therapy is pretty amazing. And again, one nice thing about NASH-TAG is it allows us time to sit back and do a deep dive into the data that's coming out. Because if you just look at the data at face value, people are looking at how much NASH resolution there is, how much fibrosis improvement there is, how much of both are we seeing. But we're not really spending a lot of time on the group that doesn't move, the groups that stay the same, and the groups that worsen. And what we're beginning to see with these NITs, and I'll highlight that again today, at least with the Regenerate data I'm going to present, is these NITs are moving in a very nice dose-response manner, even in patients that are on ordinal scoring, not changing. And they're still moving in the group that is on ordinal scoring scoring worsening. Now, it's not the same magnitude of effect of NITs, but there's still a very clear dose-response relationship. And the next several years of drug development in NASH, there's going to be a focus. There has to be a focus on what's really happening with this group that is stable, quote-unquote stable, and this group that is somewhat progressing. I mean, we take it for granted, based on the Sinacrivaroc data that came out, that 20% are going to worsen on their own and 40% are going to stay the same. We can work on the rest, but I don't think that's accurate. I think as we evolve from ordinal scoring to continuous scoring to NITs, that we're going to be able to show that there's a bigger efficacy than what we're really seeing with ordinal scoring. But I thought I would throw that out there for a conversation, because I would like to get you guys' opinion on that.
Yeah, I actually think this is really, really important point, Stephen. And I was talking to someone yesterday about this point. One of the hypotheses that I have, don't forget the bar that we're at right now. I think it's a high bar in histology. You have NASH resolution without fibrosis worsening, or fibrosis improvement without worsening of NASH. So let's take the second example for a second. When you have people that they're improving their fibrosis, and you have a liver biopsy that showed you possibly with a sample error that there's a little bit worsening of NASH, you're counting those as non-responders, versus they could be actually responders. It's just the inflammation is not gone yet, or there's a sample error. So if we started with 2-point NASH, we started without back in the days, and now the bar is higher. This is part of the group that they are improving on NITs, which look, especially with MRIs, look more globally for the liver disease itself. It's very important to give Madrigal and Intercept a credit. They need to look to break it down by the outcomes that the FDA wants, and then look at the old traditional outcome and see how it looks like comparing where we started with vitamin E when we were at 2-point of NASH and all that. I think they still have value and they're still informative, especially as you exactly said, the NITs and these groups are moving in the right direction, and over time we might see more and more improvement in this group. So some people say, well, I wish I've seen a little bit more responsiveness to these therapies. Well, maybe there is already there, but also there's the concept of combination therapy with these drugs, yada yada.
Mazen, I agree with that statement, and I'd like to repeat Stephen's toast. When he opened the meeting on the first evening, he toasted to Intercept and Madrigal, because I think these two studies are going to inform us on how to use NITs in these patients responding in the non-cirrhotic arena, and this is going to last for the next 5 years. I think it's going to be very valuable for drug development.
Thank you for that, Jörn. But I think the other thing is the AI digital pathology. I always go back to the phase 2 resmetirom data, where the ordinal scoring p-value on fibrosis was nowhere near significant. It didn't show anything, really. But when we went to AI pathology and AI digital path and looked at second harmonic generation, the F3 population, 50% of them were moving. None of the placebos were moving. And that's what helped us power the phase 3 to 60% F3 patients, and gave us confidence that there was something happening on fibrosis. And, in retrospect, looking back, one of the messages I tried to deliver yesterday was, what lessons can we glean from these deeper dives, whether it's NITs or it's AI digital path, that can help inform what's likely to happen down the road? We'll start to learn some of those important lessons, because if we just step back and look at what's being presented, we have clues to all of these phase 2s to help design the phase 3s.
I totally agree. I think we do. And at certain point, some people start saying, well, you guys are over-interpreting things with Path AI and NITs and all this. But now a day has come that, well, they are common sense things. If platelet improve, that means portal hypertension is improving. If stiffness is improving, that means there's something good happening in the liver. Stiffness reflects fibrosis, and if it improves, there's less fibrosis and there's improvement. So all that's as put together with these two studies. I'm glad many researchers kept pressing on the hypothesis and the studies that they have done, and that's where we are today. And the F3 example is outstanding, Stephen. It led to fibrosis improvement that they did not see in a non-powered study for histology previously. So it led to a great outcome in this study.
So Mazen and Stephen, we lose Jörn in about 2 minutes. I have one quick statement, which is that at some point we should take a look at where, beyond what you guys have just been saying, where AI has A-to histology is going to fit into all this. Jörn and I have a really interesting conversation with Dean Tai on that at the end of tomorrow's episode, which is sponsored by HistoIndex. But closing comment, the one thing, and we've been beating around this all morning, I just want to see if we can pin it down. One or two concepts that emerged yesterday that you think are going to have the greatest impact over the next 5 years.
I think we could continue the discussion around NITs. We didn't mention liver size. Stephen presented some data initially. To leap this all in, we can manage patients without biopsy, and I think that's a very clear statement.
Yeah, you took my thought right out of my mouth. I think we didn't spend enough time on liver and spleen volume change. I did show one separate article linking liver volume size to all-cause mortality, and it was much more predictive than FIB4. So I think
I thought that was very interesting paper. I would love to read it more. I think it's what happened yesterday. My final thoughts is continuation of what happened in NASH-TAG before. They started initially with showing MOAs and preclinical studies that can lead to efficacy, and they are here today. And I think that the discussion yesterday was focused around how to get better and smarter with clinical trial design and moving away from the biopsy. I anticipate within the next 3 years, if we can convince the regulators, especially with consortiums like NIT and Litmus and Nimble data, I'm hoping by 2026 we'll see hopefully the first trial design phase 3 that is totally NIT dependent without a liver biopsy or subpopulation with liver biopsy. That's kind of bold time and prediction, but let's see. Let's push for it.
Sounds perfectly reasonable to me, Mazen. So I have one closing thought, and then I want to thank you guys. It's been a fantastic session, and I'm looking forward to a great day today. Somebody, I don't remember who it was, in our AASLD wrap-up conversations made the point that everything that works in clinical trials over time makes its way into practice. To me, one of the really exciting things about yesterday is that while I can't tell you exactly how it's going to work, the stat geek in me is starting to see a path whereby what we're learning with all these NITs can over time simplify and create algorithms that makes it a lot easier for frontline treaters in primary care and endocrinology and screening, and then hepatology and treating, to make use of simple algorithms emerging out of the NITs we've got right now to get to some very fast, very good decisions. And I find that immensely exciting because ultimately the rubber meets the road in two places. Number one is, do we get drugs in a market? Number two is, can we use them to treat patients well? I think I saw progress yesterday on both those fronts.
For our next conversation, patient advocates Tony Vigliotti and Mike Buttel join Louise and Roger to discuss the impact NASH-TAG 2023 and the information presented here will have on patient communities and their work.
For the NASH Tsunami's team, there are 4 separate streams of positivity here. First, some great drug trials, including amplification of the phase 3 results for resmetirom and the obeticholic acid reanalysis, and an array of positive phase 2 results. Second, some tremendous breakthroughs in basic liver science coming later today and tomorrow. Third, perhaps some insights from ongoing regulatory discussions that can streamline the path to drug approvals, both in terms of time and money. And fourth, and in some ways most important of all, and we're going to talk about this a lot today, major bursts of optimism from every key stakeholder in the space about the pace of progress and the high level of confidence all this work is finally, clearly headed in the right direction. We will attempt to bring all these streams into the 3 episodes we're covering. This episode will start with a discussion with a couple of patient advocates and patients about what they've experienced over the last 2 months and what we're hearing today is going to mean for them and their work with patients, physicians, other stakeholders. Tomorrow morning, Stephen Harrison and Mazen Noureddin will join Jörn Schattenberg and me for the second half of this episode to talk about the progress with drugs and NITs that we hear in the meeting. That's going to be the episode you're hearing now. Tomorrow's episode will start with a session with Scott Freeman and Donna Cryer, 1 part basic science, 1 part overview of how it all fits together, a little bit like the conversation they had last year at NASH-TAG. The second will feature Quentin Anstey, Rachel Zayas, and perhaps 1 more key opinion leader, focusing mostly on the fireside chats tomorrow evening. And then finally, we'll wrap up the entire episode tomorrow with a conversation from our sponsors at HistoIndex, in which their Chief Scientific Officer, Dean Tai, will join Jörn Schattenberg and me to discuss a poster they're presenting here. Some really fascinating work around using AI to clarify how different drugs affect different zones of the liver, which is extremely promising for drug development as we move past just asking, does this drug work, into how does it work and where should it fit into therapy. Really exciting stuff. Monday, we'll wrap it up with a diverse set of stakeholders who will consider what have we just learned from a range of perspectives. And with all that, let's just start for today. So today we have with us Louise. Hey, Louise, how are you doing? And Happy New Year.
Happy New Year to everybody, and I'm doing very well. Thank you very much.
So on the icebreaker, everyone's going to talk about where they are and what they've been doing the last couple of days while they've been catching or not pieces of this conference. But Louise's story should be, well, actually, except for you, Tony, they're all pretty, and me, the others are pretty fascinating. So next we have Tony Vigliotti. Tony, how are you today? Happy New Year.
I'm doing well, Roger. Happy New Year to you.
And you're in Pittsburgh, which has had a week of its own, huh?
Yep.
A lot of drama in Pittsburgh this week.
Yeah, too much. wrong kind of drama.
For those of you who are not in the States, a football player, an American football player, who plays in Buffalo but is really a community icon in Pittsburgh, suffered the same kind of cardiac or on-field cardiac arrest on Monday night that Christian Eriksen suffered during the Euro 18s, if you're a soccer fan. The young man's been through a tough week. He seems to be doing better today, but it's been a major national story and a real source of heartache in Buffalo, where he plays, Pittsburgh, where he's an icon, in Cincinnati, where the game was. So happily, that's over. Or at least that's on a better path right now. And then we have Mike Patel with us. Mike is not in Canada. In fact, he's in Florida today. Mike, how are you today?
I'm great. Thanks for having me.
Pleasure to have you. So for our icebreaker, we're all going to talk a little bit about something that's happened in our lives in the last week that's a little bit out of the ordinary. Mike has a story around being on and leaving a cruise. Louise has a story about sailing, and we'll see what Tony comes up with. But Mike, why don't you do yours first, after which I'll give my lecture about cruise ships being floating Petri dishes. But you go ahead.
It does take a lot of courage these days to take a cruise. There was one last year, 800 people ended up getting COVID. Well, we had a great time, first of all, a week in the Caribbean. And then we get off and my wife tested positive for COVID. And then my son the next day tested positive. So anyway, we're getting through that. Great vacation time. But everybody's kind of used to it already, so we know what to do.
I won't give much of my Petri dish lecture because you just got into the heart of it. But I desperately want to go to Alaska. The only way to do it is by cruise. And my wife has said, great, you can go with anybody but me, and you can't come home for a week until after you get back.
Oh, hey, Roger, I do want to add, there was 6,650, I think, people on the cruise, not including staff. There was 3 people wearing masks. My wife was one of them. And she ended up getting it anyway. So there you go.
Masks are slightly helpful, more helpful to everybody else, but if no one else is wearing them, then it's not a big benefit, as you've proven, or as your wife has proven. The bigger benefit of masks is that they protect other people from your germs. Now, they protect you, but if they don't protect you as well as they protect others. So if no one else is wearing a mask, it's not going to do you a tremendous amount of good, as your wife just learned. Okay, so Louise, what's been your interesting story of the last week?
Well, I'm on a cruise, but I'm on a little bit learning to do my day skipper, which I've passed. But we've had everything from a force 8 gale all the way through to almost Riviera weather this morning in the last 5 days. So that's handy. But interestingly, in relation to Michael's story, one of the highest density COVID situations was still the cruise ship at the beginning of the COVID pandemic, was outside Sydney, I think. And through there was still one of the highest density populations of cruise. So whether it's the aircon circulating, I don't know. But cruises, when you get it, seem to be mass spread. But hopefully everybody is okay. But no COVID on our little episode this week. It certainly ticks off another first, now being a skipper of a... being able to do with my day skipper's license.
When Louise hosts the episodes this year that she's going to host when I'm on vacation, we will not call her host, we will call her skipper, or captain. Actually, Captain Campbell kind of has a ring to it, if you think about it.
Captain Campbell's mandolin, that'll be fine.
Sounds brilliant. And then Tony, so what's your story of the week?
Well, vacation seems to be the theme here. So, I have a vacation coming up. I'm leaving Tuesday for Cozumel, Mexico with my wife. And since we're sitting in 30-degree temperature here in Pittsburgh, and we've been through some below zero wind chills and so forth, I'm much looking forward to that.
So, Tony, I should note that if you want to be a truly global citizen, you're going to go from 30 to 30, right? It's 30 Fahrenheit where you are now. Ideally, it'll be about 30 Celsius where you're going to.
Yeah, that's true.
So you're not going to change the numbers, just the letter.
just a measure. Yeah.
One of the things about doing this podcast is I've had to learn how to do Fahrenheit Celsius in my head very quickly, because between Louise and Jörn, I'm the only one who talks Fahrenheit when I so choose. So I guess I've not had much of a vacation. We've just been around. We've got some personal family issues in the household that have kept everybody anchored pretty close to home. But we've had some really interesting weather, which has been nice. And I've gotten to bone up on my healthy cooking. I love to cook, and I spent the week working on healthy recipes and came up with some that I really love. So that was worth the price of admission. And frankly, if I had a vacation week where I could have been traveling, I never would have gotten to do this. So I think it's a good thing. And with that, why don't we get started? The purpose of this discussion, and Donna Cryer was going to be joining us, but she actually got to the meeting too late to make it today. She'll be joining us tomorrow instead. So what I want to talk about today, really, since I know that you two gents did not make it to the meeting, is Louise and I will start by talking a little bit about some of the things that came up in the meeting. And then I want to get feedback from you on how your work has been changed over the last month or two based on things like the resmetirom results and the obeticholic acid reanalysis and the energy and enthusiasm around that, and how you anticipate a meeting that's going to be remarkably positive will change things for you going forward. So why don't we start with the event itself? The first session this morning, and I'm not going to go into tremendous detail here, but was Harrison walking through the resmetirom data in tremendous detail, followed by Vlad Razio doing the same with the obeticholic acid data, and Lumba doing the same with semaglutide. I think the point really is that, and by the way, after break, Vincent Wong did a summary of AASLD, and I think the point is that we will shortly have 3 agents that have different but meaningful roles to play in treating this disease. The resmetirom data, it looks fairly clean on efficacy, fairly clean on safety. There is some promising emerging data that suggests there might be value in cirrhosis, and they've got this remarkably robust trial program ongoing. Vlad went through the obeticholic acid data. He went back and did the history of how we got to where we are, and then reviewed the same data that Arun Sanyal reviewed at AASLD. He went on to talk about the cirrhosis data and took an interesting point of view, which is that while that trial showed no differentiation on efficacy, no incremental efficacy for cirrhosis patients, what it showed was no decline and no notable safety challenges that you wouldn't anticipate either, which means you can put an advanced fibrosis patient on obeticholic acid with the confidence that even if it does not succeed, it is unlikely to make things worse and might have a benefit in terms of holding things from progressing further in cirrhosis. Manal Abdulmalik first pointed out on this podcast 2.5 years ago, if we could arrest cirrhosis, that would be a big deal. Or she put it today in conversation, if you take a compensated cirrhosis patient and promise me that with a drug that patient will not get any worse, I would consider that a real victory. And then in terms of SEMA, the discussion obviously centered around what we know about its ability to reduce weight and NAS scores. And then, although it did not significantly regress fibrosis over a 72-week period, why 72 weeks might be a short haul for that kind of drug, and what a valuable role it might play as part of therapy, even if it weren't the entire therapy itself. In a nutshell, that was session 1. So before we go on, I guess, Tony, Mike, Louise, in the work that you folks have been doing right now, what about any of those agents and their new data has started to affect or permeate the work that you're doing in terms of increased interest, increased energy, increased contributions, more people, anything like that? Are you feeling change yet from the idea that we now appear to have hit the target twice in phase 3 with drugs?
And from my perspective, this momentum, I think, began when I was at the liver meeting in November. Just the wave of enthusiasm and positivity over the likelihood of having a medical solution that'll help at least some people, has really just, from our perspective, just raise our urgency in getting the message out there that this is a disease you should be aware of. Lack of awareness is a big issue. It's a disease you should be aware of because, some answers are coming. I think there's still some realities that have to be faced along that path. But from our standpoint, I just think we feel an increased urgency to get the message out there.
Yeah, I have a couple of thoughts, and I agree with what Tony said. I'll go back to one other meeting, too, the Mosaic meeting that I attended. There was a lot of discussion there about the new therapies that were coming out. So 2 points. One is this is a message of hope. It offers a future. We were seeing a lot of graveyards in the past, and I hope those days are gone. And now we have something to talk about. When you think about the first line that the patients are seeing are the primary care physicians, as an example, as long as there was no therapy on the horizon, it was like, yeah, okay, what you need to do is exercise and you have to control your diet and, we'll see you again in a year. Now, at least they have to take it more seriously because these agents are coming and they're coming soon. I go back to that. Hope is a really big thing for people, and for patients, it makes all the difference in the world to know that there's something coming.
I'd echo exactly what Tony and Michael have just said. Also, what I got a sense of towards the end of 2022 was the alignment of the other specialities as well. So the potential for more patients to become aware of liver as a problem. So last year we saw the American Association of Heart put in looking for and recommending NAFLD and NASH as an assessment for patients with heart disease, published this month and released at the end of 2022, was the new American Association of Diabetes Standards of Care, whereby they advocate in recommendation 4 that patients are screened for NAFLD and NASH, particularly if you have type 2 diabetes or risks of heart disease. So I think we're now seeing that alignment, and the fact that these medications are becoming available means I think that alignment is going to get stronger. I think we'll discuss later later on in this episode some of the data that was presented with semaglutide, but I'm certainly seeing a few more people who have taken semaglutide coming for FibroScans from a different background. That's probably encouraging because I think there's a wealth of data being missed in that population because we're not doing non-invasive liver techniques. We've got a drug approved in a population where we're missing some data that we're collecting in trials now. So I think that sense of enthusiasm coupled with new medication, coupled with now a recognition within strong cohorts of patients, we might get that message out there. That offers really, really good potential through 2023 if we can get pathways in that are correct in a way that doesn't overstretch healthcare. And I think that's a big thing, certainly in the UK at the moment.
So are you also saying that there will be more enthusiasm from the multidisciplinary, interdisciplinary approach?
I think yes. Certainly what we're seeing from a US perspective, and the rest of the world do tend to follow. And I know some of our guidelines need to be updated. They're out of date. They're several years out of date, so they were probably due to be updated. But I think what we haven't seen before is this drive by cardiology and endocrinology to mention the word NAFLD-NASH fatty liver disease. We're now seeing it in guidelines. And when you certainly get the American Association of Diabetes saying in the standards of care for patients with diabetes that you should be looking for NAFLD and NASH, then we see a game changer, because that's a recommendation of a standard of care that's measurable. You are either doing it or you don't do it. That is a measurable outcome and a key performance indicator for me, even if it's only suggested and not necessarily put in the diagrams, but it's recommendation 4, I believe. So that is a game changer. Endocrinology are at the table. There was a drive at the end of last year looking at FIB4 predicting cardiac outcomes. I genuinely get a sense there is, certainly in the field, whether or not it's on the grassroots level yet, a change in momentum to join and the liver portfolio or to take control of it and give the liver disease to liver specialists. So I do think there's a change.
Roger, can I ask one more question, if that's okay? It's possible down the road that it'll be a combination therapy world, or there'll be other therapeutics coming out. However, in the short term, like Louise was saying, so if endocrinologists are using drugs like the GLP-1s, they are indicated for obesity, or they're not indicated. Are they indicated for obesity, I guess, and diabetes?
Semaglutide has Wegovy, which is an obesity drug, and tirzepatide, I believe, has or is getting an obesity indication for Mounjaro. So that would be the two, yeah.
Yeah, and so when you look at the makeup, the demographics of NAFLD-NASH patients, as we all know, obesity, type 2 diabetes are a big part of that for most people. So it's possible that those specialists might be writing the drugs for the weight side of it, and the liver doctors could be writing these new therapies as they come out, and they could end up getting combination anyway. I'm just wondering what your thoughts are about that.
I think you're right. One of my concerns is that when we look at semaglutide data, when people go to review patients who are on semaglutide for diabetes, for example, that they will not find fatty liver because they've already defatted the liver with the semaglutide. So they'll say, well, these patients aren't relevant. The fact that the drug was already working, because we don't have that pre-data, we're going to miss a section of patients in that. So I think that's a risk that we take with that. You're perfectly right. We've got semaglutide that is also, as we know, in a significant number of trials within NASH. It was a very good presentation that I'll certainly come on to later, where it was looked in combination therapy with the Gilead products. What struck me towards the end of last year in one of the podcasts was, yes, the resmetirom data is extremely pertinent and extremely encouraging, but it only ticks 25% of the population. So that means there's another 75% where drugs need to be targeted at. It doesn't mean we've solved the problem. It means we're starting to get a really good feel. And yes, you've said the graveyard may be less full. There will be still drugs, I'm sure, that fall into the graveyard. There will also be other ones that are stopped earlier in the pathway because they're not showing any efficacy in comparison to drugs like resmetirom and I suppose the beta-cholic acid, and the next ones are cabs off the rank, I suppose. So yes, you will get combination therapy. It was a big fear in hepatitis C that you would just get one doctor prescribing one drug and then somebody would add another drug without the evidence to these two drugs mixed. It's a risk, but it's also something that happens naturally in medication prescribing.
So spoiler alert to our audience, the conversation with Jörn and Dean Ty from HistoIndex and me tomorrow night, which was actually recorded the day before Christmas, will be, I think, highly relevant to this. What HistoIndex has been able to do is to clarify what zone of the liver different agents might be affecting. So, in fact, it isn't just that it's working for 25% of patients. There are specific characteristics about those patients and probably some specific characteristics about the liver, although we haven't gotten quite that far yet, where resmetirom is more effective versus where OCA might be more effective versus other drugs that come in line. So when we say we've got two drugs that each work 25% of the time, right, one of the questions is, well, have we covered half the patients? Have we covered 25% of the patients twice? And how do we know how to get the right drug to the right patient?
Maybe it's working 100% of the time in a small subset.
Right. So as we have more drugs, we'll learn a lot more about that. And I think, when Louise talks about the semi-combination data, which we'll get to in a minute, these are the kinds of things that we're starting to touch on. The interesting thing about the liver is that there are so many hypothesized modes of action because so much stuff is going on in there that it might not be realistic to think that any one drug will have a long-term effect in monotherapy on 75 or 80% of patients. We think that the efruxifermin data is pretty heroic, and that doesn't quite get to 50% on the hard measures, but it gets close, really close.
Roger, the other thing is, too, I don't think we know, because it hasn't been long enough, how long patients are going to have to stay on the therapies, too. This is not I'm sick, take this medication, and now you're cured. This is very likely going to be something that can recur.
Yeah, that's a good question, Mike, because I think, all these drugs, I know, from my own experience with post-transplant with the anti-rejection drugs, these drugs have some side effects, ramifications that become evident over time. A lot of the answers have yet to come once people are on a drug for an extended period of time. What else is going to happen to their body? So I think that's a question for the future, but kind of goes back to how long will people be on a drug? If it's a short burst, that may be minimal. But I think in all these drugs, it's not going to be the kind of thing where someone can take a pill and then walk over their couch and eat a half a pizza and drink some beer and think they're cured. So a lot of this story is still to be written.
Yeah, although the experience with statins would suggest that people are going to do that, whether it's a good idea or not. And I agree with you totally, Tony.
I suppose it all comes back to education. You don't get put on a pill or any medication without education. There was a presentation earlier on today by Suzanne Schrapton on diet, lifestyle and bariatric surgery. And lifestyle will remain the keystone of all of the treatments, whether or not you're on medication or not on medication. Because yes, there may be some side effects for some patients that they come off, or individuals. So it is a way of tailoring that, but everything's going to come with education. If you can come off a medication because you've improved your diet and lifestyle through that education process, then you would do so to reduce those side effects and to reduce any effect of that. If you then need to go back on it, whether or not that is what these medications offer us, then that may be an option. But I don't see as that being particularly dissimilar to somebody with autoimmune flares that we put on steroids every now and again to actually control the flare. To do that, there gives more scope of combination of lifestyle diet within the NAFLD and NASH community with the enhanced education that's going to come with any medication that comes out will come out with programs to locate, to educate, to tick various boxes to get access to medications. We know these are not going to be one size fits all. They're not going to be available to everybody. Everybody can have access to education and training, and that's including staff, myself, patient advocates, groups throughout the world. What can we do with these medications to enhance the way that they're distributed, the way that they're developed, and also the way that the pathway fits the communities that they are meant to serve? There's a whole network behind any release of these medications, but some of the figures are very, very good early stages. So encouragement and excitement, I suppose, for 2023.
So, Mike, one more thought and answer to your question, and then Louise, let's step into some of what came out in the sessions later in the morning and share that with these guys and get reactions. Mike, I think, we stand on the shoulders of other diseases, and everyone stands on the shoulders of oncology. One way in which I mean that here is that if you take a look at oncology, you not only get lines of therapy, first line, second line, third line, but in fact, within some of the cancers that are unlikely to completely remit, but highly likely to be managed, as in you might die with this, but you will not die from this, you wind up with the idea of induction therapies and maintenance therapies. So the simple fact that a drug might have a very, very rapid effect, but it might be injectable, it might be a large molecule, it might cost an awful lot of money, and then over time you can transition to something that's smaller molecule, oral, taken once a day, less expensive, and maybe has a broader set of metabolic effects. You're going to see therapies taking those kinds of pathways. And the better we get at taking pictures of the liver and understanding what's affecting us where, the more effective we're going to be in terms of figuring out exactly what those therapeutic pathways are and how to link them to patients. So I think over time, we will not see people taking highly toxic agents, even if any exist, for long periods of time, because if it's there to do a job, it does its job, and then you replace it with something that can maintain the progress that you just got in a less toxic kind of way.
Well, my question was less about toxicity and more about effectiveness. So semaglutide, for example, is very effective until people stop taking it. So there are some of these medications you're going to just have to stay on them, or like Louise said, if we can have some behavioral impact, then patients, when they respond, can actually be on like a maintenance approach as opposed to having to take more and more and more drugs, right?
The same thing. I mean, I'm talking about it in terms of toxicity, and you're talking about it in terms of compliance, but really people stop complying in large part because it becomes a pain in the neck to give yourself a shot and be nauseous a lot of the time. So it's not a major toxicity, but it's a convenience issue or a side effect issue. And yeah, that's right. We're going to have to craft all that over time. I have one more thing first. Mike Charlton had one amazing number this morning, which is that IQVIA ran an analysis that determined that in 2022 out of 2021, one of the recent years, that there were 200,000 ICD codes for doctor visits that included NASH in the U.S., the whole country, for a whole year. And 91% of those were primary care. And they then ran an imputed analysis based on what we know about the disease in the population. And they felt that if we were treating correctly, there would have been 16 million ICD-10 codes. So the amount of ICD-10 coding that was reported was roughly 1.3% of what it should have been. That's the landscape we're starting with. Obviously, plenty of room for growth. So, Louise, why don't you walk us through some of what you thought was interesting in the later morning sessions?
I didn't catch the beginning part of session 2, but I did catch one of the distinguished abstracts, and I suppose it was presented by a doctor called Daniel Huang, and it was about family history, looking at first-degree relatives with NAFLD and NASH, and how that related to the family line. And of course, one of the potentials of that for me is we don't want to screen everybody, because if we go around screening everybody, we're going to find various different solutions that could become extremely expensive. But we do know currently around about 30% to 25% have NAFLD. But if we're looking at liver-related outcomes, then it's about the NASH. So this was extremely interesting for me. It raised a couple of questions. They did a scoring store on the evidence they did, and they cross-referenced with the Helsinki data, was that over the age of 50, you scored a point. If you were under 50, obviously 0. If you were male, you scored a point. If you were female, you scored 0. If you had diabetes, you scored a point. If you didn't, you scored 0. And a family history, you scored 2 points. And I think the scoring grading was anything over 3. 3 or above, you were high risk. 3 or below, you were low risk. What interested me about that, and I raised a question, but I don't think it got answered, was the average age of the population, I think, was around 46. It was predominantly male, which you would expect in that age group. So it didn't show any reference to me about older women, postmenopausal women. That age group would be premenopausal on the whole, or on the cusp of perimenopause. So I didn't hear any data including on does it change as you get older for that. But I thought that was a fascinating one in the context of being able to directly relate and screen, I suppose, for me, people with a family line. We know that when you've got a liver at risk in somebody who's got alcohol cirrhosis, they tend to have children or first-degree relatives that could be considered higher at risk. Are those the groups that we should be targeting for screening as liver disease throughout grows, particularly for NAFLD and NASH? So that I thought was interesting. It was a short presentation, so I'd certainly like to see and hear a lot more about that. But it certainly gives us an opportunity, if you find one person within a family, to do a concentrated depth. And on that, I would presume if we looked at the work from the CDA Foundation, you could probably model that cost effectivity and finding and changing family diets. And I know, Tony, you've been very involved with Gina and that, looking at family diet and the family dynamics. Would that sort of resonate with you, some of the work that Daniel Huang was suggesting there?
Yeah, that's very interesting information. The question that popped into my mind was the fact that NAFLD and NASH has been undiagnosed historically, if that will become even more relevant in the future. But I think in our minds, I mean, we focus very heavily on the lifestyle side of things and actually working to prevent people from advancing to the NAFLD stage or even the NASH stage. So that's certainly very interesting data, because while we're very interested in what's going on in the drug space in NASH knowledge, we're even more interested in how we can prevent the disease from happening in the first place. And that's through education, people understanding what the liver does, how you can keep it healthy and so forth, and then what you can do in your lifestyle. In the family family aspect of it is important because the children are not the ones in the grocery store making the buying decisions. It is a family issue. That's very intriguing. I certainly like to learn more about what they were doing in their research and just getting a better understanding of that.
Yeah, and I think those were a couple of the questions that were raised. It was the case of older people tend to eat a different diet to the younger people around the table. What was at the table? All of these things that weren't necessarily part of it, but could be extrapolated or strengthened, I suppose, in the future, because we do have different diets and whether or not. So they were noticeably questions that were raised. So, but it was interesting, certainly for me, about modelling and if you're going to get down to concentrated populations, but it's also about that education.
Yeah, it's a $64 question. How do you successfully, educate the public about these things? And that's something we're constantly working on to see how we can more effectively do that.
Well, I think, you and Mike really live on the front lines of education on a day-to-day basis, right? So that is a major challenge. Mike, you were going to say.
Yeah, I was wondering in a real-world scenario, trying to apply what Louise shared from the presentation this morning, would that mean that if somebody goes to see their physician and they're identified as having NAFLD, that they would then bring in the rest of their family to get screened as well?
I think that might have been a potential. It was certainly if you were picked up with any of that, you were a high risk and you went on to screening. If you were a low risk but had other risk factors, then you were also assessed. So I think they broke that down, but I can't remember the exact criteria that they used, but the cutoff was 3 and above and 3 and below.
Fundamentally, yeah. I don't remember exactly either, but yeah, Mike, that is a point. Right now, you've got guidelines from people like ACE saying screen everybody with type 2 diabetes, but we know even there that if we put a better test than FIB4 on all of them, we would still miss a bunch of people and carry far more people into the next round than we really had to. These could become other factors that you use to get a little more predictive a little faster.
I'm thinking, too, that if you do take a familial approach when you get to the treatment stage, that you might have a better compliance because the family would do this together. We've seen that in other things, right? Like weight loss programs as an example. So I don't think it's much different than that. So not only would you have a better group to focus on for evaluating if they have disease, but also in the treatment side of it, you'd have a better success rate potentially.
Absolutely, but you also get a better acceptance. It's genetics. People don't feel so stigmatized. And you're perfectly right. You work at it together as a family. If you have alpha-1 antitrypsin in the family, you will get screened. If you're hemochromatosis, you will get screened. And then adapt the family lifestyle to benefit. And it becomes more, as you say, a team approach. And it is way more accepted. I see a number of clients who have familial hypercholesteremia and they come in to have their livers checked to then discuss with their cardiologist and that whether or not statins from that perspective, but they have their liver assessed as well. So people take to it differently. It's about targeting that liver at risk. Not everybody in the family is going to have a liver at risk, but the genetics may predispose that person's liver because of the genetics. And I think we do know that people, it's a definition then of it's in my genes. So therefore I can modify my lifestyle and let's see what can help me. And I think you do get way better acceptance and tolerability of medications and how they work. But you also get a greater buy-in. I'll only use the medication when I need it and should I need it because I'm going to adapt a lifestyle before.
The lifestyle is a key piece because that's what Tony said with the food choices too. So if the whole family's doing it, you also have a better chance of success.
Yeah, I think this is where the education of the children is really important. If they learn about it, I know going back years and years, I never wore a seatbelt when I drove, but my children nagging me about it because they heard about it in school is what caused me to wear a seatbelt.
Yeah, obviously your kids weren't rebels at that point in your life because I wind up nagging my adult son about wearing his seatbelt because he styles himself as a rebel, so he'd rather not do it. But I think this is all part of the same point, right? I mean, a lot of this is in the dynamics of family conversations, and if you get them right, it works better. We're running a little short on time, so I wanted to get to a couple of specific things that came out of the second round this morning. First of all, in Suzanne Sharpton's talk, which I thought, I don't know what you thought, Louise, I thought it was fantastic. One of the things she focused more on was what we can learn from bariatric surgery, which is remarkably efficacious, really, in terms of regressing fibrosis over time. Splendor study demonstrated that, but she pointed up some other studies that had similar results. A couple of findings that were surprising to me. Louise, help me if I get this wrong. Some proportion, I want to say 7% of the post-bariatric population developed increased consumption of alcohol that led to roughly 1 in 6 having fat on their liver that could be traced back to alcohol alcohol or to mixed disease as compared merely to diet-based NAFLD. I thought that was really interesting. I don't know if that's about if food is your addiction, you're simply trading one Jones for another Jones, or what it's about. But I thought that was really an interesting finding.
It was, and I don't think that's new data, and she was saying that. We see alcohol dependency increase in a lot of these. But somebody raised, I thought, which was a very pertinent question. Are you only now measuring the people who had alcohol as a background problem before they had bariatric surgery, or is it a genuine increase of alcohol because the others have reduced their weight and gone on and changed their diet and lifestyle as they've moved on and they've got a result from the bariatric surgery? You could argue, is it to cover pain? Is it to cover discomfort? Is it to cover all sorts of things? But that data wasn't available, so she wasn't able to draw any conclusions on that. But we do know that it's a significant problem. I think what I found very interesting, there's only 1.1% of people who are eligible for bariatric surgery actually undergo bariatric surgery. So we're still talking minority of the people who make it through that pathway get it. What she was surprised at, but I think which is very key, is that patients who've had bariatric surgery are less likely to get through a transplant waiting list program. However, they do just as well post-transplant, but a lot of them will, not a lot of them, there's a significant proportion of them will die or be delisted due to sarcopenia and weight loss and nutritional problems. So I do think there are issues, and that was more in relation to, can you perform bariatric surgery on somebody who's got cirrhosis? That wasn't your general bariatric, to be clear, that was not your general bariatric population. It was a particular cohort that was retrospectively diagnosed with cirrhosis after bariatric surgery, and then they followed them through. There was a transplant study. So, but we know that rapid weight loss can cause significant problems with sarcopenia. They did it in some of the data from the NAFLD studies. I think I mentioned it in one of the papers that was done at Arsult a couple of years ago. Increased sarcopenia and death following that. But it was an interesting one.
True that. Naeem did mention, though, and he's part of Splendor, that there's a Splendor analysis on patients with cirrhosis coming out. And I forget the word he used, but it was either good or promising to describe what they're learning. So that may have something to do there. The other place I wanted to go, and Louise alluded to this briefly a little while ago, is Naeem Al-Khoury did a paper where he talked about 3 or 4 different modes of action, all of which were basically defatting mechanism, and talked about, back at where you were a bit ago, Mike, a combination of those agents was semaglutide. And being able to get, in terms of the outcomes that we measure right now, not going into any of this AI stuff, but the outcomes, getting better effects from the combinations than you would expect from the individual agents. That putting a defatting agent on top of semaglutide for a period of time would have a greater effect than either would have alone.
It makes sense, right? The different targets. I think the important point is that we don't really know in an individual which approach is going to work the best, because I think everybody's different. So it may be a trial and error thing a little bit with some patients, and it may turn out for some people that the surgery that you were just discussing might be the effective solution. That worked well for my daughter, as an example. Not everybody wants to take medications. On the other hand, a lot of people don't want the surgery. So I think it's a choice that people are going to have to make at some point.
And it's a commitment. I mean, I also have a family member who's had bariatric surgery and has kept the weight off for years.
It is a commitment. It changes the way you eat.
And by the way, I have a family member who had bariatric surgery, kept the weight off for a while, and then couldn't. So, it's a whole new way of looking at your life. Tony, I don't recall. Did you do bariatric surgery? I know you had a liver transplant. I don't recall how you lost your weight.
No. I did not have bariatric surgery. It was actually recommended. One of the doctors talked to me about it, and I'm in that high percentage of people who were resistant to doing it. It's obviously a much more invasive solution than trying to make lifestyle changes. So I opted not to do it.
Hey, Guy, it looks like you've done a pretty good job with the lifestyle changes.
It varies from day to day.
All of us. I haven't, like I said, I've lost 100 pounds in my life. I understand that. It's not, every day is a struggle, and this hasn't been my best week, but next week will be, so it's okay.
Just to say one thing on that, some of the statistics that Naeem was discussing, some of these were 90% and 70% effect and weight and fat loss. So they were encouraging, and I think this was a combination of Gilead and Novo Nordisk, because every patient was on...
for
side effects or semaglutide.
Sema. Yeah.
Yeah, in various combinations, but it was very impressive data, the combination. So wait for those to read out later on. But nice.
Closing question. Okay, first of all, thanks to everybody for making time and for being brave and stepping up and talking about stuff that you didn't get to see, which has been, and that's been great and really helpful. The question I've been asking and everyone says, well, drugs will get approved, but that's not going to be for 6 to 12 months. So I'd like to ask you folks, from the perspective of folks who are really on the front line and dealing with patients every day, or educating patients and caregivers every day, and education and communication, what do you see the next 6 months will hold in terms of our ability to get knowledge and information out to people? And what will be your biggest aids and assets in doing that?
I'm going to jump in before the guys because I think it's going to affect them. We might see more patients or people being diagnosed with NAFLD and NASH as American Heart and Diabetes Associations rack up awareness. That drives patients who are concerned about their health to support agencies like Tony's and Michael's and GLI. There's more work coming for them to support patients. But I think the work that has already been done by all of these organisations means that they are in a way better place to help these patients now coming through. And if we can start to diagnose people early, we can bring them back. For those who are further on, we've got something to start to look at further. That's where we're going to see the changes. We're not going to see a medication approved in the next 6 months, but I think with the guideline changes and recommendations, we will start to see people becoming more aware, whether it's in primary care, secondary care and specialist care, about now having a key performance indicator that they can be measured against. So making it measurable.
Yeah, I think there's a trend that'll continue. One of the things we've seen recently as we go to health fairs and talk with the general public is that more people or know people that have a fatty liver. And I think that's a trend that's going to continue. I think that in combination with the hope of a medical solution at some point down the road. I mean, how far down the road is the question? Because I mean, even once the drug gets approved, when does it get on the shelf and when the doctors start prescribing it? I think just having that light post at kind of the end of the tunnel there I think will serve to raise awareness. So I think we're going to see increasing awareness over the next 6 months, and that is just a continuation of what's been happening over the past 6 months.
I have two that I wanted to share. I think we should not stop, even in the light of this hope that's coming. We should not stop with the prevention side of it. And so the lifestyle discussion that we had should still be part of what we talk about and what we educate. Because if we can prevent, like Tony was saying, I think that's really important. I also think that physicians, probably at the primary care level mostly, but also in the endocrinology offices, maybe cardiology, should start to actually build their be aware of their database of the patients so that they're ready. So when a drug does get approved, this is me speaking from the pharma background, you want to have the patient base ready, not go, oh, we have approval now, and then everybody's going, oh, who are we going to call? So it's, you want to know who you're going to call when the time comes, not just for clinical studies, but for the real world.
I think that's a great point. I have one thought, which is that the doctors have stated repeatedly that one of the reasons they're not engaging the disease is all they have is diet and exercise, and that doesn't work that often. I believe one of the salutary effects of having drugs coming is that you can get doctors to work harder on diet and exercise because there will be a plan B. And because, at least in the States, I would guess health plans are going to require some proof of diet and exercise on the way through to drugs.
Right. The doctor should say, show me what you can do. And when the drugs come up, I will start you first, or, I'll put you at the top of the list.
Or even doctors who aren't willing to do that, just to start working on it now, because the drugs are going to be there and this is going to be what's going to matter. I think the people who said, I don't want to hear about this because diet and exercise doesn't work very often and I got nothing behind it. Now that they've got something behind it, I think we can work a lot harder with. And that might include changing beliefs on what works in diet and exercise. We're going to do an episode on Tsunami some point in January on prescription digital diagnostics and apps, and the idea that those things work, at least to some degree, so that becomes almost a tripartite. You've got basic diet and exercise, you've got digital augmentation, and then you have drugs. And the more ways you have to approach this, I think the more people you can get interested in doing it. That would be my thought. Thanks all of you for making time to do this today. And Louise, I will see you tomorrow. And Mike and Tony, I look forward to seeing you soon. Happy New Year, everybody. Stay healthy and avoid COVID. All right?
Absolutely. Thank you for that advice.
Bye-bye now.
Thanks. Bye.
Thanks for listening to today's podcast. Please join us tomorrow when Scott Friedman, Donna Cryer, Quinton Anstey, Rachel Zayas, and others join Jörn Louise and Roger, to discuss Day 2 of NASH-TAG 2023 on another special episode of Surfing the NASH Tsunami. Have any questions for the surfers? You can send them to surfingnash.com, and we will answer on the podcast or the website.